Great. Well, welcome everyone to this webinar entitled Digital Pathology and Artificial Intelligence 101, Unlocking the Value from the AP LIS. My name is Eric Walk. I'm a pathologist and the Chief Medical Officer at PathAI. And it's really my pleasure to be part of this webinar with my two colleagues from PathAI, Jim Sweeney, President of PathAI Diagnostics, and Dr. Sean Kinsey, Medical Director of PathAI Diagnostics, as well as Ed Youssef from NovoPath, Chief Strategy Officer there at NovoPath. This is a webinar that we're partnering with NovoPath on. And as you can tell from the title, the goal is to update you all both on digital pathology, but also with a focus on the value of the interface between the LIS and digital pathology. We can go to the next slide, Jeff. We do have some legal disclaimers that I want to show briefly. If you could put that one back, Jeff. I'm not going to go through this all. Feel free to screenshot this and read it later. I do want to mention that the information presented today is for general purposes only and should not be construed as professional or medical advice from PathAI, PathAI Diagnostics, or its affiliates, or any of the presenters. And also some of the products and services that we'll talk about today are RUO, for research use only, and are not intended for diagnosis or treatment. Okay, now we can go forward. So here's our agenda for today. You can see we have 8 topics to talk about, all around digital pathology and LIS. I'll provide a brief context setting and introduction to digital pathology. Then I'll move in to the topic of drivers and value drivers of digital pathology and AI. I'll then pass it to my colleague Jim, who's going to talk about the framework for how to evaluate vendors in the digital pathology and AI space. I'll then give a brief overview of PathAI, and Ed will give an overview of NovoPath. And then I'll come back and talk about the importance of the interface between the anatomic pathology, LIS, and the IMS. Then Dr. Kinsey is going to give a very interesting case study describing his experiences of going digital and embedding AI in a real laboratory environment. That's the PathAI Diagnostics Laboratory in Memphis. And then we'll conclude and hope to have time for Q&A. So just to set some context, I think it probably is very evident to everyone on the call, but digital pathology and AI pathology is definitely having a moment with tremendous interest and growth in companies. And it seems that wherever you go these days as a pathologist, you're encountering the topic of digital and AI pathology, which of course is a good thing. And there are many ways to observe this trend. On the left here, you can see from a publication perspective, the incredible growth in the number of publications on digital and AI pathology. And if you look at this data, there are a couple inflection points in the 2010 era. There was an inflection point around the availability of technology like high throughput scanners. And then more recently, certainly the COVID pandemic triggered a lot of interest and momentum in digital approaches. On the right, you can see, I'm sure you've seen a lot of these press releases and announcements between different companies and organizations. That's another sign. And also, if you've been to a pathology meeting lately, I'm sure you've seen the tremendous activity and interest in digital pathology. I was just at the CAP meeting, and before that, the European Congress of Pathology. And I would say if there was one theme of both of those meetings, it would be digital pathology. The next slide. Another data point that you can look at is just the number of companies in the space or entering the space or the ecosystem of digital and AI pathology. And I'm not going to go through all these, obviously, but to break it down, there are really 3 categories of players in this evolving ecosystem. You have the scanner companies, you have image management software companies, and you have AI companies. And yet another way to look at this is on the next slide. Now, these companies and different institutions are partnering with each other. So it's hard to keep up with these partnerships. So this is an interesting way to visualize this from DeciBio, where you can see it is a bit of a tangled web of partnerships. And it'll be really interesting to see how this plays out in the future. I think some themes looking at this graphic are that a lot of the partnerships are being driven by scanner companies, AI companies, the IMS companies, but also academic institutions and large labs are participating in these partnerships. So with that as a brief context, the purpose of the webinar today is really to educate you on the field of digital and AI pathology, but with a focus on this important piece of the LIS and how the LIS interacts with digital pathology and the IMS. So just in the way of some expanded introductions, again, my name is Eric Walk. I'm Chief Medical Officer at PathAI. I'm a board-certified pathologist, but I've spent the vast majority of my career in the industry, both in drug development with Novartis and diagnostics development with Ventana and Roche Tissue Diagnostics. And today I'm CMO of PathAI, overseeing the medical team, including 25 pathologists in our Memphis pathology lab. Jim, I'll turn it over to you for a brief introduction.
Good morning, everyone. Jim Sweeney, I'm the president of PathAI Diagnostics, and pleased to be here today and share this webinar and our experiences with our journey through digital pathology and now introducing some AI models. Okay.
Thanks, Jim. Sean?
Hi, I'm Sean Kinsey. I'm the laboratory director of PathAI Diagnostics, and it's been an interesting journey going digital and now deploying some algorithms. So I'm happy to share this information with you.
Thanks, Sean. And last but not least, Ed.
Thank you, everyone. Thank you, Eric. Ed Youssef, Chief Strategy Officer at NovoPath. I've been with NovoPath for about 17 years now. I started as a developer, a software engineer, and now, throughout my experience and history with NovoPath, I'm really excited to be at a time where I can see technology really making an impact on the pathology market.
Thanks very much, Ed. So let's continue with our agenda. We'll move to the second topic, which will be drivers of digital pathology and AI. So again, I don't think this will be a surprise to anyone on the call, but there are some challenges in the practice of pathology today. And I have no doubt that not only are you aware of these challenges, but you may be directly impacted by these challenges. Reimbursement, for one. We all know that reimbursement continues to shrink. We all think that's unfair, and we continue to fight for that, including the CAP, where I'm very active, has multiple advocacy activities ongoing to stop the reimbursement cuts that we see as a regular occurrence every year. In light of the reimbursement cuts, it's particularly painful that the volume is increasing. Increasing volume is a good thing, of course, but not in the setting of decreased reimbursement and decreasing resources, which we'll talk about in a second. But I talked to a lot of labs, and they are struggling with that good problem to have of having more specimens coming in, more testing needing to be done. So that's definitely a challenge. Pathology resources. The graphic you're seeing there, I just pulled from Pathology Outlines. If you go to the jobs section, you'll see this curve. So Path Outline tracks the jobs open for pathologists. And you can see, and you probably feel this yourself, it's hard to hire pathologists. There's a shortage of pathologists. So if you look closely, that graph is saying there's 700 available jobs for pathologists right now. And you can see that's a dramatic increase. There was a little dip after the pandemic, but then it rocketed up. And we speak to a lot of labs that, including our lab, that struggle with that of how to find pathologists for their practice to meet the increasing demand. And last, but certainly not least, I'm calling this category information overload. Pathologists are being asked to do more with less. So more tests, interpreting molecular data, keeping up with the literature, keeping up with FDA approvals and guidelines. So it's really becoming quite overwhelming for pathologists in the field to integrate all this information into the pathology report, to channel it to the clinicians and to the patients who need to benefit from this information. So there are absolutely some headwinds facing the practice of pathology today. So moving to the next slide, the hope is that digital and AI pathology can help with some of these headwinds and deliver value to the lab, both in the form of efficiency, but also quality. So here on this slide are some value drivers that can be realized just with regular digital pathology. And by digital pathology, I mean scanning a glass slide and viewing that slide on a monitor. And we'll talk about AI next. And I think these value drivers are probably very familiar to many people. Collaboration and remote consultation, probably many of you are doing that already. Workflow efficiency and reduction of turnaround time. I think Jim later on will talk about our experience in our lab in Memphis, digitizing slides and the efficiency associated with that. Slide preservation, we all know that slides can fade, both IHC slides and H&E slides. So digital locks the quality in at the time of scanning. Diagnostic quality. I believe Sean will talk about some of the basic pieces of value that he realized just by going digital, including being able to instantly pull up historic cases instead of having to run to the slide room or the archive. The ability to view multiple slides side by side, for example, multiple IHCs and a lymphoma workup. And then something called trail tracking, where many digital systems allow you to see which parts of the slide you've looked at and which parts of the slides you haven't looked at yet. Cost savings. So I think Jim also will talk about the cost savings and the ROI that we benefited from in our lab in Memphis just by going digital and not having to send so many slides via courier. And then finally, on the pathology demand side, can digital pathology be used to improve the recruitment and retaining pathologists? So in our experience, I'm not sure if Sean's going to talk about this, but we've hired several pathologists that are fully remote and sign out their work digitally. So DP could potentially help with the pathologist resourcing problem. On the next slide, we have now the AI side of things. So beyond what I just told you about, how can AI add incremental value drivers that will make the adoption of digital AI pathology cost positive or revenue positive? So one is decreased turnaround time and increased throughput and productivity of pathologists. And this can come in many forms with AI. We'll talk about some of these later in the webinar. Things like AI-enabled quality control, tumor detection in an automated fashion, the quantification of biomarkers like PD-L1. If you look to the right, that is a study that we conducted using our PD-L1 non-small cell lung cancer algorithm. This was a study of 10 pathologists across just about 500 cases. And what we saw is a pretty dramatic reduction in the time it takes to sign out PD-L1, which may not be surprising for those of you who sign out PD-1, because you know that involves counting and estimation of 1% cutoffs, et cetera. Going back to the left, so can AI increase the competitiveness? We've heard a lot of labs bring on AI and think about the quality improvements and the efficiency improvements in a setting of differentiation. So how can they use AI to differentiate themselves from their competitors in the lab space? And finally, new revenue streams. I think this is a value driver that many people are not aware of. And a lot of this can come from biopharma. So biopharma today is really struggling to enroll patients on trials. There are literally not enough patients to enroll in all the trials that are in clinicaltrials.gov. So any way to expand the enrollment pool, they are very open to. And this could involve AI, where, for example, we enable labs that don't typically enroll patients on clinical trials to do that with things like molecular prediction algorithms from H&E slides. So next, I'm going to pass to Jim, who's going to talk about a framework for how to evaluate vendors in the digital pathology and AI space. So over to you, Jim.
Okay. All right. First of all, I want to introduce you to PathAI Diagnostics. This is a video that I was going to quickly play, and then I'll talk about some of the uniqueness of our facility. And on the left, you can see very quickly some of the key highlights of PathAI Diagnostics. First of all, for those of you not familiar, PathAI Diagnostics is formerly known as Poplar Healthcare, that's based here in Memphis, Tennessee. And Poplar Healthcare has been around since 1995. We service the areas you see there in outpatient pathology. The first one was obviously GI, which the company was originally called GI Pathology. Today, we service over 1,500 clinicians across the U.S. We have a strong partnership with FedEx. We do over 2,000 cases a day. We're known for quality work, first and foremost, with our expert pathology team, fast turnaround time, and extensive payer networks. The other thing I want to quickly mention to you is that the journey that we began with digital pathology actually began in 2018. We made a conscious decision then that we wanted to get involved in digital pathology, and we embarked on an extensive program and analysis that was actually led by a pathologist. We'll work closely with our business development team and looking at the landscape and beginning to understand all the key players, whether they were the scanning companies, the emerging image management system companies, and where algorithms and AI companies stood at the time. Next slide, please. So today, this is a brief summary of where we stand, and I'm going to go through after this a little bit of the analysis that how we got here. But today we currently use 3 G2450 scanners and 1 Hamamatsu to deliver slide images to pathologists around the country and also here in the building. Our scanning today, we're scanning over 30,000 slides a month, and you see that we have validated, and this is probably a couple of weeks old, but we have validated 9 pathologists, 6 fully. What that means is that they strictly read on a digital basis. And as Eric's already pointed out, and Sean will comment, some of our more recent recruiting of pathologists, it's now easier for us in this challenging recruiting environment to find pathologists and have them work remote, where we can then set them up to be validated and work with us through our system. Next slide. All right, so what did we have to think about? Well, there are 4 main things that we needed to really understand, and not just understand today where things stood, but where were they possibly going to evolve into the future. The first was our current, at that time, LIS system. How strong was it? Was it capable of being able to engage with digital pathology, and how would it interact with digital pathology? So there was a learning curve there. The second was the whole slide scanners, image management system, and AI, and I'll go into a little bit more detail on each of these to tell you the thought process. Next slide. All right, scanners. You see here a chart that on the one axis tells you the cost, and then on the other axis the throughput. And in that upper right-hand corner, you can see the more expensive ones obviously have higher throughput. You see where we have the G2450s and the Hamamatsu and where they fall in. Below, you see all the questions that we had to ask as far as throughput, uptime, speed, and then also interoperability, reliability, and then where were these scanners in their life cycle? How quickly could they possibly be supplanted, i.e. the AT2 we knew at the time was eventually going to get phased out, which has subsequently been announced. So we wanted to make sure we were investing in the current technology and in the future. To be honest with you, we worried less about FDA approval, because we knew that with CAP, we could do a LDT validation, and that's how we've entered into the market. And we obviously focused initially on primary diagnosis. So you see, those are some of the key thoughts that we had to go into the scanners. The next slide, please. Then when we thought about image management systems, the key here was, and at the time I used the word agnostic. I wanted to be able to not just use the scanners that I had in our facility, but also be able to receive images from other labs around the country that had perhaps different scanners. So it was important for us to pick an IMS that was agnostic to scanners and could obviously interact and work with any of them. And on the flip side, on the other side of the coin with AI, we knew there was a ever emerging business with AI companies that were introducing different algorithms or beginning to explore the use of algorithms. Some were diagnostic in nature, some were based around companion diagnostics. And we wanted to make sure we took all of that into consideration. So you see down below a lot of the questions that we went through and that we asked ourselves as we were evaluating this. Next slide. All right. And then the last but not least is storage. Okay. And before I actually get into storage and how we first launched and how we operated, one of the key things that I didn't appreciate and that I got a quick lesson and learned from our IT team was bandwidth. Okay. Did we have the ability with our internet connection, since we were going to be doing two things? One, initially working with an IMS system that was outside of the building. And then secondly, we would be working with clients that we were transmitting our images to. So we had to make sure that we had the capacity. Today, we have actually two 10 gig circuits that flow in and out of the building. One is obviously there for backup, but it was a key analysis or a key part of the analysis that we went through to make sure that our internet and bandwidth capability could support what we were doing. Now, if you're obviously doing this internally within your own building and scanning and just sending within the building, that might have a different analysis. But I just wanted to call that out and share that with you. What we initially did was we would store the images for 90 days, after which time then the images were erased. That was driven by cost because of the storage cost associated with maintaining these images over a long period of time. Today with PathAI, that's different, but it's one of the things that you can consider and think about, because obviously you have the glass. No one has replaced the requirement to store and maintain that glass for a cap for 10 years. So our thinking was we could always go back and rescan that slide if necessary. All right, next slide. Okay, and then when it comes to AI, all right, again, when we were thinking and looking at with the image viewer, as well as how we wanted to enter the marketplace, primary screening was our main focus. We had a number of clients that we were servicing through business models, PC models, that we wanted to be able to digitize. And then as we looked forward, we wanted to understand where was AI heading with workflow enhancements, all right, what you see there on the left-hand side, and then whether AI was going to be applied diagnostically or with companion diagnostics, which is ranging from that second category of diagnostic assist tools all the way to the biomarker quantification or quantitation. The other offshoot that this has created for us is we are now heavily involved from a histology perspective with companion diagnostics. And we have grown to become a partner with many of the IHC companies as they're evaluating new technologies. And we're pleased to be one of the labs that are in these early access programs and being able to introduce them, some of which will have AI tools associated with them, and others that are just, at this point in their early stage of development and then being looked at in the initial rollout to support the decision-making when it comes to different therapies.
Thanks very much, Jim. Great overview. So to continue on that theme, I wanted to provide an overview of PathAI and how does PathAI fit into the framework that you just heard Jim describe. So looking at this workflow between NovoPath and PathAI, we cover end-to-end the entire value stream within digital and AI pathology. NovoPath, obviously, they are a leading provider of the Pathology LIS, and you'll hear from Ed next on some of the details around their solution. And for the rest of the components here of the ecosystem, PathAI covers these either directly, for example, the image management system, which is called AI Site for PathAI, as well as the AI algorithms. And the other components like scanning and data storage, we cover through partnerships. So again, between NovoPath and PathAI, we're really covering the end-to-end entire framework that Jim described. Unfortunately, we won't have time to do a demo of AISight, which is our image management system, but I invite everyone on the call to contact us if you're interested in a demo, because we would love to show you in detail how the system works, including the algorithms. But in a nutshell, you're seeing the two main components on the slide here. On the left, you're seeing the slide management area of AISight. This is where a pathologist would select cases, search for new cases, organize cases into folders, etc. And on the right is more the work screen, where a pathologist could either view a case digitally for sign out or apply the embedded AI algorithms that I'll talk about in some detail. And incidentally, what you're seeing there in the screenshot is the HER2 algorithm, where the algorithm is showing you the score as well as the heterogeneity of HER2 scoring, if that exists. So in that particular case, you're seeing a heterogeneous case that has some HER2 1+ areas in green, but also some HER2 2+ areas in blue. From a regulatory perspective, we have multiple versions of AISight. One is RUO, and another, which we call AISight DX, has been FDA 510(k) cleared and CE marked for primary digital diagnosis. And 6 features to remember about AISight are in this slide. First and foremost, I think the most important is that AISight, our image management system, is AI native. So this is meant to be the central hub for all your digital pathology work, whether that involves AI or not. But when you do need AI, that's integrated directly into the IMS. You don't have to click out to another system. So that's very important. And we'll go into some detail on exactly how that works. Number 2, and this is a word that you've probably heard a lot in the DP space, interoperable. And specifically what we're talking about is that AISight supports all major scanners and all major LISs. And I'll also talk about a product that helps with those interfaces called AISight Link. Third, rapid activation and scale. So this is not on-premise software that needs to be installed and managed. So this is really cloud-based and pushed through a web browser. So our browser is Chrome. So your work exists in a Chrome browser tab. So it's very easy to activate and scale. The entire solution is cloud-based, meaning that the compute for the AI occurs in the cloud in a scalable way, and the storage also exists in the cloud. So making it very easy for end users to bring the amount of storage and compute that they need. Open algorithm access is becoming very important. So in addition to our algorithms that we develop at PathAI, AISight is able to integrate third-party algorithms. So if you're at an institution that has developed an algorithm and you want to integrate that into our system, we would love to talk to you about that. And last but not least, iterative development. So we have a very aggressive release cycle. So every 6 weeks, we are iterating the platform with based on feedback from pathologists, so adding new features that pathologists are asking for, which is very important. On the next slide, I briefly mentioned AISight, and so this is the architecture map for how AISight interacts with LISs like NovoPath and also scanners, and it's really AISight Link as a system of what's called application programming interfaces, or APIs, that really serves as the translation device, if you will, between AISight and these other parts of the infrastructure. So very important. And in a few slides, I'll go into some detail on how that works in a bi-directional way. So now to transition into more the AI part of our solution. So I want to describe some of the algorithms that we've developed. And these exist in 3 categories: research and biopharma translational, workflow efficiency for the lab, and then pathologist assistance for the laboratory. So starting on the left, we have a lot of exciting research applications that you can see there. Explore is our branding for the research application. So we have Path Explorer for the tumor microenvironment, IBD Explorer, NASH Explorer, et cetera. And the common theme behind all these Explore algorithms is the generation of a new type of biomarker that we call human interpretable features, which is essentially morphology-based spatial biomarkers that one can use to discover new correlations to endpoints, diagnoses, treatment effect, toxicity, et cetera. Molecular prediction is another research grade application that we're developing. The idea here is, can we use H&E algorithms to predict the presence or the absence of a molecular alteration, like an NTRK fusion or an ALK fusion or EGFR mutation? And the thought here is that we could help with the process of identifying cases that do or don't have these changes, and then follow it up with a confirmation. Under workflow efficiency, these sets of algorithms have really resonated with pathologists for the reasons I talked about earlier on the challenges in pathology. So auto QC is a good example. So we have a very sophisticated artifact detection model that automatically identifies folds in the tissue, out of focus areas, even ink marks and dots on the slide, because obviously you want to address those things before the diagnosis is done. Tumor detect, also really very powerful. So it does what the name implies. It automatically detects tumor. And why would you want to do this? There are two immediate reasons. You might want to sort cases based on the presence or absence of tumor. So you can sign out cancer cases first. Also, as part of the NGS workflow, most labs are using human pathologists, taking the time to estimate how much tumor presence is on the slide as a way to increase the success rate of NGS. So can this be done automatically with AI? We think so with tumor detect. And then lastly, pathologist algorithms. So one category that we're heavily involved in is disease severity assessment for diseases like NASH, ulcerative colitis, lupus, and celiac disease, where the commonality here is that there are very subjective and irreproducible scoring systems where AI can help the reproducibility of these scoring measures, especially on clinical trials where these measures are used for endpoint assessment and enrollment. And lastly, biomarker quantification. So I can't see hands on this slide, but I would virtually ask how many people like scoring PD-L1 and counting those cells. I did this recently at the CAP meeting, and in an audience of 200 pathologists, and not a single pathologist raised their hand. So can AI help take away those tedious tasks and assist pathologists by counting cells for a TPS score? The same for HER2 and HER2 low, and ER and PR and K67. So we feel that this is an important category of value for AI going forward. We can go to the next slide, which I think is an expansion of what I just showed you. Just visually, you can see our menu of algorithms. The only thing I'll add here is that you'll see both for PD-L1 and Tumor Detect and PathExplore, we're developing these algorithms across multiple histologies. And this is very important because when you develop a, say, tumor detector for lung cancer, I think it's obvious to everyone that algorithm would not work so well in colon cancer or renal cell. So each of these has to be developed separately, and it's a strength of PathAI that we have 550 pathologists on our contributor network that work with our machine learning engineers to develop these tools. And to give you a sense of what it takes, each of these different indications will take hundreds of thousands of annotations by a human pathologist, so very time consuming. But we feel it's very important to get the performance that's needed. Lastly, I wanted to show you a bit of a visionary slide on how AI could add value to the workflow in histopathology. So really focusing downstream of digitization. So we talked earlier about the value of having digital slides. This is really focusing on the value of AI. And the common theme to all of these applications is that you're using AI to pre-process the slide in advance of the pathologist doing the case review. So how much information can we provide to the lab, into the pathologist in advance of the review to make that review higher quality, but also faster? So some examples: reflex IHC ordering. Could we have the AI look at, for example, all the breast cases overnight before the pathologist looks at them and automatically order a HER2 for those breast cancer cases that are positive, but not for the negative cases? I briefly talked about tumor sufficiency using tumor detect, and also this molecular prediction idea from H&E. So these all fall into this theme of doing things in advance. Auto QC and restain I talked about. So could the AI, in our case, the artifact model, automatically order a recut or a rescan when the quality was not sufficient? Intelligent case sorting. Can we use AI to triage cases to different people in the network per specialty? And biomarker quantification I've already talked about. So there's a lot of value that I think we're just in the early phases of discovering of how AI can add efficiency, add quality to the process of pathology and the sign-on of pathology cases. So now I'm going to pass it to Ed, who's going to do a NovoPath overview.
Thank you, Eric. So we'll do a quick highlight about the NovoPath 360, which is our flagship, a product and the LIS of the future. NovoPath 360 is a cloud-native SaaS LIS, delivers Class A security, scalability, business continuity, also offers a depth of functionality that meets the complexity of the pathology workflow of today, while embracing the latest technologies of the available for the future as well. So we are partner focused on meeting the evolving needs of a AP market, and we come alongside our labs to ensure that they have all the tools and the technology that really can deliver the efficiency and accuracy to their diagnostics. So to focus on today's subject, if we can go to the NovoPath slide, we have that. Yep, thank you. Focus, back to our today's subject. A NovoPath offers an out-of-the-box seamless integration with your LIS, with your whole slide imager and AI solutions. So everything that you've heard from Eric previously around how AI can revolutionize your lab and your diagnostic process, NovoPath can offer you an out-of-the-box a solution that kind of integrates and puts all that together for you. So you can configure and you can maintain your AI algorithms and all that out of from NovoPath, so that they're working hand in hand together. The seamless integration will allow the pathologist to work from a single system. Also allow your admins to work in a single system, configuring the workflows. And it ensures the security and accuracy while your pathologists are focused on the diagnosis and the digital images. So as a pathologist moving from one case to another or from one slide to another, NovoPath will automatically work in the background to switch your cases, make sure that you're looking at the correct slide, since you don't have a physical slide, managing that the connectivity and the communication between the LIS and your IMS or your AI solution. Additionally, NovoPath 360 can offer an AI-driven slide distribution that will deliver a mechanism to drive these slides or deliver those slides to be read by the pathologist. That can increase the efficiency of the lab operation and also improve the diagnosis turnaround time. So looking ahead to the future, just to highlight a few of the crucial benefits of choosing a system that is well integrated. Having your LIS and digital AI system integrated will definitely give you benefits from the ability to automatically trigger available algorithms to aid and speed up the diagnostic process. We'll also bring in the results, the AI results, back to NovoPath, store that with your case, and available to review at any point in the future. Depending on the final diagnosis, you may also wish to include that in your final report, and that will bring in or will drop in all the required disclaimers along with that. We're finally working we're constantly working with our partners to make sure that we're accounting for all regulation changes when it comes to digital pathology. So we know there will be a lot of changes in the future. And as much as possible, we'll try to account for that and be ahead of the game, whether it's coding, billing, or other regulations that may impact your LIS or how you how you design your final reports, or how you report your final results. So with that said, I'll turn it over back to Eric, who'll go more into the importance of having an integrated AP, LIS, and IMS system.
Thanks so much, Ed. Yep. So I do want to double click on the topic that Ed teed up, this idea of integrating the IMS and the LIS. Here is a survey showing the importance of this in the minds of end users. So this is a survey where people were asked, what are the most important criteria for selecting an IMS? And the LIS integration topic was highly ranked, just behind cost and UI user experience. So very important in the minds of end users. So on the next slide, you can see how this would work. So what is the value in connecting the LIS and an IMS like AI Site? And I think Ed touched on this, but to go into a little more detail and try to give you an example of how this would work. And again, with AI Site, we have a sister product called AI Site Link that manages this interface. So for example, at the time of case creation, all the demographic data information on the specimen gets transferred through AI Site Link to AI Site. So AI Site has that information because no one wants to manually input demographic information, of course. Then at case review, again, flowing through AI Site Link, one would launch AI Site directly from the LIS, like NovoPath, and view cases in AI Site. Then when it comes to AI, if AI is relevant for that particular case, the AI algorithms, as you've seen, are embedded into the AI Site IMS, and then the data would go back to the LIS through the bidirectional interface. So we go to the next slide, you can see a mock-up of what this would look like using NovoPath as the LIS. So one would click on the case within NovoPath, that would open AI Site, you would do your work in AI Site for, this is a HER2 example, and then let's just say it was a HER2 3+ case, then that 3+ score, then would go back over the interface back into NovoPath. So once this solution is in place, I think you can see that there are a number of benefits. So it's very simple, it's seamless, so it's transparent to the user. So your users are not managing this. They're just clicking and then everything is happening automatically. It's flexible, meaning our systems people would work with you and your specific IT solution to make sure that this fits for how you're set up. It's scalable, meaning if your lab grows to other sites, then this can grow with you. And most importantly, it's secure. So we are compliant with HIPAA and GDPR and managing personalized healthcare data. So just to expand on this further, here's another way to look at this. So here you're looking at the workflow, starting with accessioning, glass slides, scanning, an AI piece, and then pathologist review. So it's an animated slide, yeah. So here, and bidirectional is important. So just to explain and give an example of bidirectionality. So going in one direction, the LIS delivers the case level and slide level metadata to, for example, AI Site, where the AI is done. Then if we click again, yeah. So then once the AI environment is active, then we can do some of those workflow streams that I was showing before. For example, the auto QC, the restaining or rescanning, that could automatically be triggered back to the LIS. And then in another example of pathologist case sign out, the pathologist could click, as I've shown you, open the case from LIS into AI Site. And then if we click one last time, then let's just say it's a PD-L1 case, then the TPS score that was generated within AI Site then goes back bidirectionally into the LIS. So both directions of communication are very important for this truly to be seamless. Okay, we can go. Next, I want to pass it to Dr. Kinsey, who's going to describe a very interesting case study in Memphis involving both digital pathology and AI. So over to you, Sean.
Thanks so much, Eric. So again, just kind of reemphasizing some points that have been previously made. AI Site is both an image management solution and the viewer all in one. And the image on the right, I see quite often when I'm looking and evaluating my cases. The image on the left, the image management solution, it's kind of working in the background. I actually don't see this screen very often unless I'm searching for a case specifically, but my LIS actually functions, generates this list for me. And when I double click on my cases, that's when the image viewer changes. So if we go to the next slide, I think the common question I have is, how do you get started, and what does that look like, and what is the validation process? So for the validation process, it's actually kind of complicated for your pathologist, because they have to look at least 60 cases, and they look at those 60 cases on glass, and then at least 2 weeks later, you're going to look at those same cases digitally, and going by the guidelines that CAP has generated. On the right side here, you can see what my form looks like when I do a digital validation for my pathologist. Some of the notes that I make as we're doing these scores, are immunos used, are special stains used? If there's a difference in the diagnosis, keep in mind, we're not looking for correct diagnosis. We're actually looking for equivalent diagnosis. Did the pathologist make the same diagnosis they did 2 weeks ago? So that's what my form looks like. And a couple of the blanks on the top, I think, is important, because if any of these change, CAP and CLIA are actually asking for a new validation. Did the viewer change? Did the monitor change? Do you have a second monitor that you use at a second location if it's a different model? And of course, the scanner. So if these things change, because those are essential to the actual image itself, we actually do a full new validation for each pathologist. So we can go ahead and go to the next slide. So once we got started, there were some benefits that I saw as a practicing pathologist. Some of the ones have been mentioned a few times today, is our reduced costs with both couriers and shipping. The other one that's so helpful for me in our practice is I can actually have pathologists that don't live in Memphis anymore that practice under our CLIA. I'd like to show the bottom screen, as a busy pathologist, I have several slides, probably about 20% of my cases each day. I'll order a stain or a deeper or a combination thereof. And each morning when I get my stains and deepers, or in the afternoon in a lump, I spend a lot of time rearranging my slides. Now with the image management system, that's all done for me automatically. And I didn't realize what a huge time saver that was. And I practice primarily gastrointestinal pathology, but my DermPath colleagues, the number of stains they order is actually much higher. And I actually have a full-time employee that her entire job is sorting these slides and giving them back to the pathologist in the correct order once those stains are complete. Now that job is becoming reduced as my dermatopathologist switch to digital pathology and how they practice. So my remote pathologist, okay, with the last slide, please. I'd like to also comment on consults for my remote pathologist. In previous years, we'd have to package up that case, send it to them. If they had a question, they'd actually package it back up and send to us. Now with AI site, what would take days now takes seconds to minutes. They highlight an area they have a question on. They send it to their favorite or least favorite pathologist for their consultation, and that pathologist will respond within minutes. And it's really helped efficiencies in a way that I wasn't anticipating with our practice. The fourth box here that I think that all practicing pathologists dread is when a clinician calls and questions our diagnoses and asks for us to re-review something. Previously, that would mean that our staff would have to go to the storage room, find that slide, bring it back to the pathologist. We'd have to review it, then call that clinician again. Now, most of the time, when I get these phone calls, I can actually retrieve these slides while that clinician is on the phone and share the slides with them or talk them through it. And something that I will admit surprised myself is that I am just as fast and efficient in my diagnostic time as I was on glass now that I am digital. We'll go to the next slide. I think my favorite feature is what we're looking at here, the days of stacking slides on top of each other to see if our B cells are co-expressing different markers is over with this side-by-side view, and it allows me to zoom in and pan uniformly. It's just a beautiful feature that I never thought I would love as much as I currently do. So there's some of the features I've found as a practicing pathologist that I think are so beneficial. If we go to the next slide. Then going from digital, our first algorithm that we validated is the AIM-PDL1 assist tool. For that validation, I used 61 cases and I used all 4 clones. And I treated it very much like I did a validation of a digital pathologist, where I had that 2-week washout period between when I initially evaluated slides and then when I evaluated using the algorithm to help me come up with these scores. And there was an excellent concordance between my scores and the algorithm. But there were 3 cases I'd like to highlight that actually had a poster at CAP this last week that I highlighted some of my own deficiencies. So eating a little humble pie here, I can tell you that there were 3 cases out of those 61 in which I gave a score of 0 to less than 1. And with the algorithm's assistance, I actually changed my score to above 1. And it really helped me identify some low positives, where those critical thresholds where we make clinical decisions. This is where this algorithm has been most useful to me. So if we go to the next slide, I can walk you through what this algorithm looks like on my computer. This is a PDL1 stain. And if I click on the overlays and go to the next slide, you see there's all these color changes where it highlights the positive cells and negative cells that are cancer. And it also highlights immune cells and the stroma and areas of necrosis. And we can go to the next slide where it actually gives me the values. And you can see that score that it came up with of 7.61%. That's something, depending on your level of integration with your LIS, like NovoPath or some other brand, that you can get that incorporated directly into your LIS result form. Of course, you have to validate this and make sure that you agree with the stain. I think it's interesting that it tells me exactly how many cancer cells there were, 40,000. It's pretty impressive. So we take a closer look at this and go to the next slide. I think it's very helpful to look at this on a cellular level, and we can see individual cells are marked here. And then once it draws my eye to the positive cells, I can go to the next slide and look at this without the overlays, and it shows me where the positive cells are. Go to the next slide. And again, here's just those low positive cells that I could have potentially have missed. And since this algorithm was deployed, we've actually released a second algorithm that we use now, helping us with some liver diagnoses and quantification. And we go to the next slide, and I think we could probably spend another hour talking about what we could do in the future. The possibilities are absolutely endless, but I'll leave this slide up here. And I think that brings us to our conclusions. And we'll probably open the floor for some questions and answers in the last couple minutes that we have here.
Thanks so much, Sean, for that great case study. So we're just at time, so I think we'll skip the conclusions and just try to get to at least one question. The theme I'm seeing is around billing. So maybe Sean or Jim, when a pathologist is purely digital, where do they bill out of? The primary lab? If not, does the offsite location require credentialing from insurers? And then related to that, relative to your statement, we have pathologists that don't live in Memphis anymore and practice under CLIA. Are you billing from the CLIA site?
Right. All right. So that really falls into 2 main buckets. First, if it's our pathologist, meaning our employee, and they're working remotely. So for example, today we have somebody in New Jersey and someone in North Carolina. We bill out of our facility here under our CLIA and NPI. They're credentialed through the plans here in Memphis. And of course, there's a disclaimer that goes on the report indicating that the work was done through digital pathology as validated as an LDT. If it is a TCPC model and we're shipping glass out to a pathologist in California or Florida, and they're employed by another practice, whether it's a physician practice or a pathology practice, they would obviously bill accordingly. And in most cases, they're either co-located in that practice or they're affiliated through their CLIA.
Thanks, Jim. And then maybe we'll end on one question for Ed. will you elaborate on the AI algorithm that you are alluding to that is native to NovoPath 360 that will address case distribution? What does the back end of that look like? What technologies partners are using to build that AI, for example?
So that's a NovoPath-driven algorithms that we've created in-house, and that takes a bunch of different rules and factors into the distribution calculation. So we can look at the pathologist schedule, their skills, the skills required on the cases that to be assigned. We also take into account client preferences, the availability at the time for the pathologist, and we load balance that across all the pathologists within the pool to figure out which pathologist is best fitted to work on that case. We can also take into account the stat cases, rush cases, priority. So all that work together within the rules. We play that in different times throughout the day so that it's constantly the NovoPath from the back end trying to figure out the best pathologist for each one of those cases that needs to be read, and make sure that we're always maintaining that TAT expectation to get those cases out of the door as quickly as possible.
Thanks very much, Ed. So I know we're past time, but I'm not sure if people can stay on, but maybe we'll try to squeak in one last question. This one's on storage, so maybe for Jim. How long did PathAI keep digitized images prior to getting into AI, and how long do you keep the images now?
Hi, this is Sean Kinsey, and I'll actually address that one.
Okay. Thanks, Sean.
Prior, we kept images for 3 months before they were deleted. Now, because we are part of PathAI, we keep these images indefinitely to help us with our model development. And it really doesn't have anything to do with the AI component. It was a cost-saving endeavor for us before we were part of the PathAI group.
Yeah, great. Thanks, Sean. Okay, I'm going to lie and do one more question. So are there any examples in the pathology ecosystem where the LIS and the IMS is developed by a single company? I am not aware of that, and I think that's where this interoperability and all the partnerships come from. So I don't know if Sean or anyone else on the call has another view, but I don't see
I can tell you that when we were exploring going digital, we did not identify any companies that could do everything with both an image management system, a viewer, and an LIS. We were looking for that, and that solution did not exist at that time.
Yeah, maybe that'll happen through some consolidation and acquisitions in the field, but as of now, I don't think there is a single source for everything. Great. I think we'll end it there. I want to thank everyone so much for joining us today for this webinar. I want to thank Ed Youssef from NovoPath, my colleagues, Sean and Jim from PathAI Diagnostics. I hope you found it valuable. Please follow up with us if you have additional questions or you would like to see a live demonstration of anything that we have shown today, because we would love to do that. So thanks, everyone, and we'll see you next time.